One of the things that I'm really interested in is how the brain changes at a cellular level. In order to examine that, we do a lot of immunofluorescence on our Leica microscope here. And what we're typically looking for is how patterns of activity change following distinct learning events. So when animals are learning about environment A or environment B, based on their prior history with those environments, what's really going on in the brain and what's changing in response to that. And you can see distinct sort of patterns of activity throughout this brain by taking pictures of it zoomed in really, really far, so about 200 times of what you normally see on a slice. And we can look at distinct populations of cells and their neural activity associated with each event that we're interested in studying. This microscope is interesting because it allows us to take sort of wide field view of exactly where we are in the brain. So right now I have about 10 slices all within the same field of vision, and what I can do is click around and just navigate, almost like using Google Earth to click around a different neighborhood. So I can say, from a really broad perspective like I have here, I'm interested in looking at, say, the dorsal hippocampus. What I can do here is click, and I see where the dorsal hippocampus is here. Click, the microscope will go there itself and take a fine-grained detailed picture at something like 40x or 20x, whereas right now we're just looking at a 5x. And what this is really interesting about this is what I can do is have these pictures taken concurrently so that we're getting a whole bunch of data all at once. This allows us to map the entire brain in a way that on previous microscopes and other typical fluorescent scopes would have taken months to do. I can do it in a matter of days here.